Hello, welcome to another video from the conscientious biologist Ben Gallagher. This is the fourth and final one in the genetic engineering playlist, and it looks at bacterial engineering. Specifically, we're looking at using bacteria to create human proteins. Now that can be incredibly useful because there's lots of conditions where humans can't make one specific protein, and that can be very detrimental to their lives. If we look at diabetes, for example, and type 1 diabetes, people with that condition can't produce their own functional insulin, so they need to inject insulin taken from another source. Now, that source is normally bacterially derived by genetically engineering bacteria to get them to produce human insulin protein that can be harvested them to be given to diabetics. We're going to look at the process by which that happens. This is from the GCSE syllabus, and as always, please do subscribe to my channel if you haven't done so already. Thank you. So given everything we've looked at so far in genetic engineering has been engineering of eukaryotes, mostly animals we've talked about, but we've given a few examples of plant engineering. It's worth just going back and looking at the two main types of cells in life, which is prokaryotic and eukaryotic, or prokaryotes and eukaryotes. Now, prokaryotes, we're really just talking about bacteria. Eukaryotes is everything else. That's the four main kingdoms of life: animals, plants, fungi, and protists, or protista. Okay, that's what we've focused on. We're going to look now just at bacteria though. So let's quickly recap bacterial cell structure. Now, like all cells, they've got the two essential components for being a cell, a compartment. They've got the membrane, which is the skin around it, and they're full of cytoplasm. So you've got a bag. Once you've got a bag, you've got a self-contained little unit, which is a cell. Of course, cells must be able to make proteins because the proteins then do everything, if you remember back from so many lessons. So they've got the DNA and they've got ribosomes that can read the DNA. Now notice the big difference here. The chromosome there in the bacteria is a loop, okay? It's referred to as a circular chromosome. The chromosomes that we've looked at in everything else so far have just been linear. They've been one strand. Remember, when that goes through DNA replication, that one strand makes a copy to form the chromosomes that have that X structure, a double chromosome, as opposed to a single chromosome when it's one strand. Go back and look at the cell lessons, specifically the one on mitosis, if you're not quite sure what I'm talking about, because that's what explains the single and double chromosome thing. But in bacteria, we have this circular chromosome. It's a loop. So the DNA is different. But the code and the nucleotides and how the DNA works is exactly the same. Now, bacteria can also have this going all the way around them. This is a cell wall. This provides them with strength and protection. And if you remember back to the videos on osmosis, one of the major functions of a cell wall is to stop water going into too much and bursting the cell. Remember, our cells, animal cells, if you put them in water, pure water, water will move into the cytoplasm by osmosis, the cell will swell and it will burst. That doesn't really happen in bacteria because of that cell wall preventing them bursting. Exactly the same as plant cells. Bacteria can also have a flagella. Not all of them do, but a flagella is a projection. It's a protein projection from the membrane that can move about to allow them to swim, either towards favourable conditions or away from unfavourable conditions. A lot of bacteria also have this slime capsule going around them. That's a mucus secretion that they put out around to try and keep themselves hydrated and protected. But where we are really interested when it comes to genetic engineering are these little things. These are little extra loops of DNA, like extra but smaller circular chromosomes that bacteria have. Those are the useful little bits of DNA that we can use as genetic engineers to manipulate some new genes into those bacteria. So let's look at those plasmids in a bit more detail and compare them back to the lesson we did on human DNA and on the structure of DNA. And you're going to see a lot of similarities here. Now, if you haven't yet watched my video on the structure of DNA, I would really strongly suggest you do before you carry on watching this one, because it's really important you understand exactly the structure of DNA and how it works before you can look at this. So go and look at that if you haven't done so already, but I'm going to assume you have, so let's carry on. So if we're looking at plasmids, they are made of DNA, which means they're going to have the same basic nucleotides as our DNA. They've got C, G, T, A, cytosine, guanine, thymine, adenine. Now, of course, DNA has to be double-stranded. So those nucleotides will have ones that complementarily base pair to them. So C goes with G, A goes with T. So you get this double-stranded arrangement, exactly the same as our DNA. Now, of course, that carries on. But in a plasmid, the DNA bends and starts to turn. So you can see you can get angles in it. It's not kind of straight like our DNA. And that can just carry on and make a complete loop all the way around. So we get a perfect loop of double-stranded DNA. But you can see this is basically identical to our own DNA. It's a double strand of DNA. Now, this gives genetic engineers something small and simple to manipulate. You just need biochemical scissors and glue. Because think, if you're going to manipulate this, if you're going to put something new into it, you need to be able to snip it open, put your gene in there, and glue it into place. Now, thankfully, there's enzymes that can be used for exactly these jobs. There's two specific types of enzyme that can cut DNA open. Those are called restriction endonucleases. You don't need to know that term, but it's a nice little bonus term to throw in there. So you can use restriction endonucleases or restriction enzymes to cut, and you can use another enzyme called ligase to glue new bits of DNA in. If you imagine you were doing this with paper, if you had a loop of paper and you wanted to stick a different coloured piece in, you could snip open your loop with some scissors, put your bit in and put a couple of bits of sellotape on to hold it in place. That's essentially what we do with genetic engineering of plasmids. Right, so this is the really important slide. This is where we go through the process of how you do that genetic engineering to end up with a gene product like insulin. So the diabetics have got insulin injected into themselves to control their blood sugar. So if I start with my bacteria over there and I put the plasmid into it, and you'll notice this time I've used the proper DNA double-stranded plasmid that we drew in the last slide, just to link it up for you. Okay. Now I need that plasmid to engineer, so I'd remove that plasmid from the bacteria. I've only made it bigger for scale so you can see it doesn't grow. Okay, but I take that plasmid out of the bacteria. What I now need is a source of the insulin gene that I need. So I take someone, anyone, it doesn't matter. We could just say that this woman who isn't a diabetic, so we know that she has the correct gene that can be read to make the insulin protein, the correct insulin polypeptide that folds up to the correct shape of the insulin protein. Remember, with proteins, it's their shape that controls their function. So this is just a non-diabetic person. Now we know that inside her cells, there must be that gene. So if I take one of her cells, and if I can identify and remove the insulin gene, which I'm representing here just very short, but remember actual genes would be hundreds and hundreds of nucleotides long. I've just made it really short there for simplicity. But if that purple bit of double strand up there, if that's my insulin gene, I've now got hold of that. Now what I need to do is now take my plasmid, use my biochemical scissors, my enzyme, cut it open, so that I can then slot my insulin gene in there, use the other enzyme to glue it in place. So if you now look at my plasmid, I've recombined the bacterial plasmid with a human gene. So it's called a recombinant plasmid because I've recombined different things. So I've genetically engineered a recombinant plasmid. It's bacterial DNA, but it contains a human gene. And look, my purple bit of DNA matches perfectly with the bacterial DNA. It's double-stranded, it's using nucleotides, it's the same bases, it's the same code. Now what I need to do is get that recombinant plasmid back into a bacteria. Now that bacteria won't know that that DNA doesn't belong to it. It doesn't know it's got some human genes in it. So that bacteria just thinks it's its own DNA. And that bacteria is going to do what all bacteria do, which is multiply. And the bacteria multiply by something called binary fission. It's not mitosis. It's a similar process in that it makes exact copies and clones, but it is slightly different. So binary fission, that is a key term that you need to know. And by binary fission, then one bacteria becomes 2, becomes 4, 8, 16, 32, 64, and so on. It just multiplies until you would have thousands and thousands of bacteria, all of them containing your insulin gene, because they all contain the plasmid, because when they did binary fission, they made exact identical copies of themselves, including the new plasmid. So all of these cells should now contain your plasmid, which contains your human gene. So they're all going to start making insulin proteins. Let's say these purple blobs are representing the insulin proteins being made and secreted by those cells. So there's loads of insulin now, just in whatever solution you've got your bacteria in. Now, if you imagine you're keeping those bacteria in an enormous vat, a huge, great barrel, okay? Inside that huge barrel, you've got the liquid that they're growing in, that you've added nutrients to and oxygen and everything else they need. And those bacteria are just multiplying away happily, releasing their insulin into the solution that you're growing them in. All you then need to do is kind of scoop it out and purify it, filter it to remove the insulin proteins, put that in bottles, and we've got human insulin. It's literally that woman up there, it's her insulin from her gene that's been created, but made en masse by these bacteria, which can now be purified, bottled, and given to diabetics. It's a very simple, very clever bit of genetic engineering. And this is actually decades old. This has been going on for about 40 odd years. Really, in terms of the whole thing, quite an old bit of DNA engineering to get this done, but a brilliant process. Take a screenshot of this one. This one tends to come up quite often in the exam, so make sure you really understand this process. And that's it for genetic engineering. That's all 4 videos that hopefully you've watched on the genetic engineering topic. From here, I would suggest you go on to the disease topic. Links up quite nicely, given we've just been talking about diabetes. So head to that one next, please. As always, please do subscribe to my YouTube channel if you haven't done so already. Like this video if you found it useful, and head over to my Facebook page for some extra bits of biology, just to add some depth to your understanding of the entire curriculum. Thank you.